Welcome to ISL Enabled Virtual Lab of CSIR.
So, welcome back, everybody. We now have Dr. Ramrai Bhatt, who is a cancer researcher from Indian Institute of Science, Bangalore. He studies how cells become cancerous. So, he does a lot of experiment with actual tissues, but also uses computer software to decode how cells behave. So, with that, Jigyasa, I think we can go ahead and ask our question, shall we?
So, Dr. Ramre, why do cells all of a sudden become abnormal?
This leads one to think about what is what is normal for a cell and what is abnormal for a cell to be in. And one needs to understand that normal or abnormal for a cell can be looked at not just at the level of a cell, but more in terms of its relations with its surroundings. What makes cells normal is when the relationship of these cells with their other neighboring cells and their matrix is normal, when they can speak to each other and communicate with each other through signals, chemical, physical, and so on, and then decide that, okay, together we are in a state where we are able to function, and therefore we don't need to necessarily do two things. One is move and two is divide. So in healthy organs and healthy tissues, therefore, what you have is that the organ or tissue remains stable, by which I mean that the cells of that organ or tissue don't go wandering around, don't start dividing either. Over time, these relationships between the surroundings and the cells can be altered due to various reasons. These reasons could be, for instance, risk factors that come in through environment. You mentioned things that happen on the surface of our skin. So ultraviolet radiation, injuries, things like that, food habits, risks, usual things, aging, smoking, so many other risk factors that we are exposed to, are stress in our lives, and so on. All of these confounding agents basically start disturbing that relationship between cells and their surroundings. And ultimately, when those relationships through those communications are disturbed, that's when the state moves from what I said, don't divide, don't move, to either divide or move or do both, depending on the extent of that miscommunication or lack of communication.
So, how do you actually study such relationships between the cell and the environment, and what causes it to change and so on?
There are various ways by which you can study this. Again, experimental cell biology and experimental tissue biology uses various models and or various systems to study this. We, for instance, try to take the extracellular matrix that I talked about, the other cells, the normal cells, cancer cells, put them together. It's like almost like doing Lego. We can put whatever we want together under a microscope and then try to study, does the cell divide? Does the cell move? Or does the cell remain stable? And we see that for a lot of cases, for example, when we take normal cells and when we put them under the normal environment, the cells remain healthy. However, if we take normal cells, not cancerous cells, but put them within an environment which is not typical to them, which is not what they would see inside the body when they are in an animal or inside us, they would actually start dividing and growing. So it tells you that in a lot of cases, what is normal or not normal, therefore, is guided by the environment in which you would be in. What we study in our laboratory, or what most of us study, as I'm talking on behalf of sort of the community that works and so on. We've been studying. We invariably study cancer cells from both humans as well as from animals, which act as good model systems. We believe act as good model systems for the cancers we get. For example, mice are commonly used as model systems because in many mice we do get the kind of cancer that we also see in humans, and so mice have also been studied, and murine cells, which are cells from mice, have also been studied to look at how cancer occurs and so on. However, However, we now realize that it's very important to for now that we know quite a bit about cancer, quite a bit about human cancers. We've reached a stage where it's being increasingly important for us to study most of it with human cells. So that requires us to actually go to patients make cohorts of patients from different places, take their cells and grow them. So that's the reason why, yes, we do take cells from biopsies and so on.
You said you use microscopes. So I'm sure that there are certain kind of advanced microscopes that you might be using. So maybe you can tell us what kind of microscopes these are and what kind of detail that you get to see with these microscopes.
Some microscopes allow us to look at an entire tumor. So if I'm interested in looking at a population of cells and how a population of cells behave, I use microscopes that give me a more zoomed out view. On the other hand, I want to look at the mitochondria inside a cell in a cancer, which is present in the middle of a bunch of cells. Then I would want a microscope which would focus me there at very high resolutions. So I would use, say, a extremely high resolution confocal or 2-photon microscopes and so on. I can use another microscopy to actually not even see. So most microscopes see, but some microscopes feel. So I can use atomic force microscopy, for instance, to feel a tumor rather than see a tumor.
I realized that you were also a clinician before becoming a developmental biologist and a cancer biologist studying development of cancer. So we were curious to know about the transitions that you made, a couple of them.
When I did enter a very good medical school in Calcutta, and I was enjoying my first year when I was exposed to topics like anatomy, biochemistry, physiology, and so on. But as time went by, I realized that I was in a sort of a dilemma as I moved more and more deeper into medical school. I was stuck in a dilemma as to as to whether the disease and how the disease occurs and how the disease spreads or takes place, was that more interesting to me, or was it more interesting to me to understand, okay, now that I know that it has happened, how do I diagnose it and how do I cure it, which are extremely important questions. But I was more interested in the more fundamental questions. So I very quickly realized that I have to sacrifice one if I have to pursue the other. I was actually studying limb pattern formation using chickens as a model. So that's one of the classical models of studying limb development. So that's what I did for my PhD, and it had a lot of very interesting dimensions of both evolutionary biology, or how developmental mechanisms evolve, and as well as of physics, where how physics of form plays a very deep role in guiding the shape and size of organs and tissues. So it was, like I said, as unmedical a problem as possible.